Well, hello everybody. Today we're going to be talking about the first part of Chapter 2, which is on the subfield of biology and behavior, which encompasses neuropsychology, which is what I am, a neuropsychologist. So I obviously find this chapter very interesting. This is very biologically based. There are a lot of terms and a lot of information, which is why we're going to split this up into two separate sections. So in terms of a case that kind of unifies this chapter, your book is going to be talking about Brandon Burns, who was serving on the front lines of the Iraq War and had a bullet pierce his skull and tear through the back left side of his brain. His treatment was surgery where they had to remove part of his skull and a third of his brain. And this will come back to how this case is important in that how we use people who have had different types of brain injuries to figure out what part of the brain is important for what types of functions. And that was particularly many years ago, but even to this day. And also the idea of neuroplasticity in terms of how do we recover from brain damage? Can you recover from brain damage? Can you recover if 1/3 of your brain has been removed? So we're going to start with a brain quiz. Oh, don't worry. It's not a quiz for any points. But just to see what you know about the brain, here are 8 questions, and we'll have a few of these throughout the semester. So what I want you to do is just read the questions and write your answers down on a piece of paper. You could pause this right now, and when you have your answers completed, then unpause it, and I'll show you what the answers are. So go ahead and pause this now. All right, and now here are your answers. So we'll see how much you knew about the brain, that there is no sense of pain in the brain itself, which explains why neurosurgeons probe areas of the brain while the patient is still awake. That's true. The adult human brain weighs about 3 pounds. The first month of life, the number of connections between brain cells dramatically increase, and that was also true. The average adult has approximately 100 billion brain cells. That's a lot. Neurons, which are the cells of your nervous system, transmit nerve signals to and from the brain at up to 200 miles an hour. The right side of the brain controls the left side of the body. Oh, here's kind of a sad one. By our late 20s, the first signs of brain aging can be seen on brain scans. Unfortunately, that's true. Once you've reached your late 20s, it's all downhill in terms of your brain, which is why we need lots of brain researchers to help keep our brain in great function for many, many years. And regular mental exercise helps protect against Alzheimer's disease, and that is true. That's the use it or lose it principle. So keep using your brain. All right, so the nervous system is a communication network that conveys messages throughout your body by using an electrical and chemical process. So we talk about brain and neurotransmission as an electrochemical process. There's an electrical component and a chemical component, both of which we'll talk about today. The brain consists, as I said, about 100 billion nerve cells, 100 trillion connections, and provides the power to think in all the complex ways that we do and differentiates us from most other organisms. And the brain is really the last frontier of scientific inquiry. There is so much we do not know about the brain. And if you take a physiological psychology class or a neuropsychology class, we end up with a lot of answers of, we don't know. We just don't know. We're just starting to understand how the brain works. So neuroscience would be the study of the brain and the nervous system, and integrates fields like medicine, computer science, and psychology, trying to figure out how the brain works. The brain is kind of unique in that if you just look at it, you can't really see it working. Different than if you were to look at a heart, you can see your heart pumping blood in and out, and you can kind of figure out how it works by looking at it. the brain doesn't do anything that we can actually see with our eyes. So we really are trying to figure out how the brain works. And then biological psychology focuses on how the brain and other biological systems influence human behavior. And this is a subfield of psychology, biological psychology, neuropsychology, kind of all in the same area. So how do we start to study the brain? It's been a long progression. It started in the late 1700s with something called phrenology. This was first proposed by Franz Gall, who tried to explain functions of the brain by bumps on your skull. And here's a phrenology kind of mapping. And so if you were to feel your skull right now, you would feel that there are some bumps. It's not perfectly straight. There are some little bumps, some little indentations. And so Gall made a map and would say, hey, if you're feeling in one particular area, you can kind of read these numbers, and you have a bump, let's say, I don't know, over here, this is 7. You got kind of a bump on that side of your skull, then 7, that would mean you're very secretive. And so, he would feel all around your skull and try to tell you, depending on the bumps on your skull, what kind of characteristics you would have. Now, this has proved to be absolutely not accurate at all. However, it was the beginning of someone trying to connect your brain, because obviously what's under the skull but the brain, and behavior in some way. So it was the beginning of that. Also, in the kind of early 1800s, we started learning about how the brain works by ablation, which is destroying parts of a living animal's brain and then determining whether some functioning was lost. So take mice or something like that and damage a particular part of the brain and then see, after we damage this part of the brain, now the mouse, can't use his right paw. So that would mean that that part of the brain is important for that. Or you damage another part of the brain and say, now that animal has doesn't eat anymore and is not hungry. Okay, so that must be somehow involved in hunger. And so that was another way we learned about the brain. Now, recently, probably the last 50 years or so, we've had great advances in imaging and brain recording technologies to allow us to observe the brain in living human people as we do different things like sleeping, reading a book, telling lies, and we can see what's going on. And so now we've got these really great ways to study the brain. And we break them into 2 categories. We can look at brain structure. What are the parts of the brain? And the 2 main ways we look at brain structure is computerized axial tomography, or a CAT scan, you might have heard that term before, or a magnetic resonance image, or an MRI. Those are really the ways we look at the structure of the brain. And you can see here, this is a picture of a CAT scan, and this is the brain. This white is the skull around it, and you could imagine if we were looking down through the top of somebody's brain. So we're looking right down through the top, kind of middle of that person's brain. And computerized axial tomography is basically an X-ray. Many of you have had X-rays before. That does multiple cross sections of the brain. And like I said, here we're looking at the top. These are called ventricles. These are holes in our brain. They are supposed to be there, and they allow what's called cerebrospinal fluid to go through your brain, which is important for keeping it healthy. CAT scans are very inexpensive to do. They're very quick, just like if you've had an X-ray before, it goes very quickly. And you can see major structures or significant damage pretty quickly. Now, you can contrast that with an MRI scan, and maybe some of you have had an MRI before. An MRI is a newer technology. It uses a magnet instead of X-rays, and creates this very strong magnetic field that passes through the brain, and the computer can analyze the response of the brain cells, basically, and the fluid in the cells to create images of the brain. These MRI scans will also allow you to see structures, but as you can tell, it's much more detailed. You can see many, many more finer details on an MRI than a CAT scan. And this is also looking at the top, looking down from the top to someone's brain, pretty much the same area. And you can even see what we call the gray matter on the outside here, the white matter, and many more detailed structures. Now, MRIs take a lot longer. If you've had an MRI, for my research, we've had to give many MRIs, and I study children. So to little children, they can take up to 45 minutes, an hour, and you have to stay very, very still, because just like any picture, if you move, it's blurry. So that can be difficult, especially when you're talking about studying children, or if you have a real emergency situation, you have someone who just came into the hospital after a car accident. Well, you might want to do a CT scan first to see if there's any major bleeding or damage that you can see quickly. And later on, if the person is still continuing to have problems and you're concerned about brain damage, you may decide then to do this more in-depth MRI. MRIs are also much more expensive than a CT scan. This is a picture of how it looks to get an MRI. You kind of lay down on this table and it moves you in, and there's this big magnet that takes a picture of your brain. Again, just looking at a CT versus MRI, where the CT doesn't give you as much detail as this MRI scan does. Here's an MRI. This is the side view, so you can see the person's profile here. And you can really nicely see all the different structures of the brain, and we'll talk about some of these, the corpus callosum, the cerebellum. You can see all the bumps and grooves of the cortex. You can see here's the hindbrain, the spinal cord going down. So really beautiful pictures. Now, if we wanted to see the living brain in terms of its function, how it works, we can also do that. And we've got a few different techniques. We can use an EEG. An EEG basically has, people get these electrodes that are kind of glued onto your scalp. It doesn't hurt, it's non-invasive. They just kind of stick all over your scalp. And then each one of those electrodes records the activity, the electrical activity that's going on in your brain. And the computer then can read out what's happening in each part of the brain. And then you need to be a really expert in reading EEGs to be able to say, oh, and well, this person, maybe she's reading a book, or we're asking her to do math problems, or we're asking her to tell a lie, and we can see which electrode, which part of the brain right under that electrode is particularly active. So that's one way to study brain function. fMRI, probably some of the more commonly used today for looking at brain function. This takes the MRI scan that we were using, but it adds what we call a functional component, functional MRI. And in this case, they take the same MRI picture using a magnet and then are able to look at the flow of blood to areas of the brain that are working. So again, if we're asking somebody to do math, then the areas of the brain that are working hard to do math would have extra blood flow going to those regions. And the fMRI scanner is able to pick up on the flow of this oxygen-rich blood and will make these little colored areas that say, oh, this part of the brain is working really hard. This part of the brain is working very hard. And this part of the brain is working hard as well. And so you can see what part of the brain is working when somebody's doing a task. Of course, the key is the person can't be moving, so they have to be very still. So something that you can do lying down, we often use video screens or something like that where we ask them to look at pictures or think about math answers or things like that. Another way we can look at the function of the brain is using a PET scan. This is positron emission tomography. In this case, you have to have the patient get an injection of a radioactively labeled substance, that's called a tracer, that's injected into the bloodstream, and it's tracked while the patient performs a task. And then the computer produces this image of the brain that shows which areas are most active. Those are the areas of red, orange, and yellow, to the areas that are less active, that's the greens and the blues. All right, but of course this one requires an injection of this radioactive substance, which is relatively safe, but often people would prefer an fMRI if possible. This was an interesting article from Scientific American article called A Great Attraction. And this is another way that we're using in terms of technology to maybe affect the brain or change the way the brain works. This is magnetically stimulating the brain, could lift depression and perhaps even boost creativity, but questions remain. And this is just a little subsection from that article that when this psychiatrist stepped into an elevator in a London hospital in 1990, he had no idea the ride would transform his research career. A fellow passenger was having a giggling fit for no apparent reason. When George inquired about the outburst of merriment, the man replied that a doctor had held a magnetic coil, that's what this is, just a handheld magnet, against his head, and it made his thumb twitch uncontrollably. Even though it sounded a bit like quackery, George was curious. He contacted the doctor, who said he had stimulated the man's motor cortex in his brain, located at the top of the head, in hopes of seeing whether it would spark any signals to any muscles. So in this way, this is a non-invasive kind of treatment idea that we can actually affect the way the brain works by using this relatively safe magnet on the outside of your brain, on the outside of your skull. Kind of an interesting thought, and we're going down that way for research in terms of ways to treat maybe depression or different psychological and psychiatric disorders. All right, so let's talk about neurons. What are the key structures of a neuron? And a neuron is a cell that your brain and your nervous system is made up of, and it's specialized to communicate with other neurons through an electrical and chemical signal. So first, let's just look at the structure of the neuron. This is a neuron right here, and it has a few parts. It has this circular part. This is the cell body. It has dendrites. These are those branching features that are coming out of the cell body. It has an axon, which is this very long kind of tube structure. And then the axon terminal is at the very end. So let's take a look. Here's another picture of a neuron. We'll start with the dendrites. These are those branching fibers that come out of the cell body, and these are responsible for getting information from other neurons. Imagine there are other neurons all around here, and the whole point is to send information, right, from one neuron to the other. It's almost like a game of telephone, if you played that when you were younger, where you've got to like send a message from one person to another. That's what these neurons have to do. So imagine there's lots of neurons here, and these dendrites are almost like hands or arms reaching out to get information from the neurons that surround it. And I'll explain to you how that information is transmitted soon. But right now, just know that these dendrites are these branching fibers that are reaching out to bring information into the cell. Then we get to the soma, or the cell body right here. This is what keeps the cell alive, very much like all the cells you probably learned about in biology. It has the nucleus, the mitochondria, ribosomes, other structures that are found in cells. Now, after that, we have this axon. This is the yellow part down here, this little thin fiber that sends information, right? The information came from another neuron to the dendrites, into the soma, and now we're continuing to send a message all the way down the axon. And then the axon terminal, there's a lot of names for this. There's also called the presynaptic terminal, but I think your book calls it an axon terminal. That's just the end of the axon. And so that is the structure of a neuron, where we've got dendrites. This is actually an actual neuron through a microscope, the cell body, an axon, that long thin fiber, and then the terminal, the end of that axon right there. Now, of course, in real life, it's much more complicated. Your brain has 100 billion neurons all smushed together in your skull, and so they're all all tangled together, and it's hard to really tell one apart from the other, but they are, in fact, individual cells. There is one other type of cell in your nervous system called a glial cell, and these are cells that support, they're basically a support system for the neurons. They help provide nourishment, they protect the neurons, they, in some cases, provide myelin, which we'll talk about in a few minutes, which covers the axons. Glia means glue in Greek, and so they really are the glue that holds the nervous system together. They take away things that might potentially harm the cells. They're good at getting waste, taking waste out of the area where there are neurons. And there are many, many more. They're much smaller, as you can see, but there are many more of them. So there are fewer neurons. The neurons are really doing all the important communicating. And then we have these many, many more small glia that are keeping the system running smoothly. There is research now, lots more research on glial cells, thinking that they maybe do a lot more than just hold things together. They may have very important things that they do on their own. And so they're getting a little more attention now, which is very cool. All right. So we talk about communicating within neurons, and what are we talking about when we say send a message? And when I say we have to send a message, it gets a message from these other neurons that comes in through the dendrites, and then we have to send this message all the way down the axon. How do we send the message all the way down the axon within a neuron? So I'm not talking about how do we send a message to another neuron. I'm talking about sending the message down this axon. And these axons can be quite long, because that's the whole point, is to send a very message from a long distance, right? Imagine you stub your toe and you've got to get that message up to your brain. So there is a neuron in your toe, and it may have the axon go all the way up your leg, which, depending on how tall you are, could be 3 feet long, right? Maybe longer than that. Probably most of you are taller than me. So this can be a long axon. We've got to send a message through. So how do we do that? And that is an electrical process. Okay, so communication within neurons is the electrical part of the electrochemical process. And in a simplified way, and again, if you take another, like a biopsych class, you'll learn more about this. The neuron is surrounded by fluid. There's fluid all around our body, all around the cells of our body, and the fluid has ions. Maybe you remember the word ion from high school chemistry. Ions are charged particles. And the charged particles there are more positively charged particles in the fluid. That's this purple stuff, is the fluid. And here is the axon. We're looking kind of inside of an axon, like a cross section. The axon, you can see these negative signs are more negative compared to the fluid. So the fluid is more positive, and the inside of the axon is more negative. Okay, and so this axon, this yellow axon here that we're looking through, imagine it would be like a cylinder, it's enclosed, but it is what we call selectively permeable, meaning that some things can get in, while some things can't, right? What does permeable mean? It means things can get in or out. And if you had something that was totally permeable, like an open door, anything can come in or out. If something was not permeable, like a closed door, nothing could get in or out. And selectively permeable, maybe something like a screen door, or screen on a window, let some things come in, right? Very little particles of dust or tiny little insects, but not other things. And so that's the case on this axon membrane. It's selectively permeable. Some things can come in and some things can come out, and that's pretty key. When the cell is at rest, which means it's not sending a message, nothing is happening. The cell is just hanging out, doing nothing. It's at what we call its resting potential. Makes sense? When it's at rest, it's at its resting potential, which is negative 70 millivolts. You just have to remember this number of negative 70 millivolts, which means the inside of the cell is more negative than the fluid surrounding the cell. And if you want to know how much more negative, negative 70 millivolts. Okay? Now, what happens when we want to go from rest to action? Like, it's getting a message and it needs to send this message. So a neighboring cell would stimulate it. And again, we'll talk about how that happens in a second. But let's just say the cell next to us got a message and now it's sending the message to us. We're the neuron next door. And it's saying, hey, we got a message. What happens? Channels start to open up. That we said this is semi-permeable. Channels start to open up. And that's here in section B. So A is this same picture, right? We're at rest, nothing's happening. And we are, if this is, this graph here is time along this x-axis and millivolts along the y-axis. And we're at negative 70 millivolts over time. Nothing's happening. We're just resting. We've got more positive on the outside, more negative on the inside, and we're at our resting potential of negative 70 millivolts. Once I get a message from a neighboring cell, channels in the neuron's membrane start to open up. And so that's happening here in B. We get these little openings. And when we get these openings, because the outside of the cell was positive and the inside is negative, and again, maybe you remember this from chemistry, opposites attract for ions. And so all these positive ions that are out here want to go inside because the inside is negative, right? The inside was negative 70 millivolts. So all those positive charges start going into the cell. They're very attracted to the negative ions that are, or the negative charge inside the cell. And so once those channels open up, the positive ions start moving in, the positive charges start moving in, and you can see what happens. If you have positive charges moving into the cell, we started at negative 70 and we are getting less negative or more positive, right? We hit negative 55. And then we're even going all the way up here to 0. And then it actually gets to be positive inside the cell because so many of these positive ions rush in or flood into the cell because they are so attracted to the negative charge that was in the cell to begin with. So positive ions enter the cell, and there is something called a threshold potential. So resting potential was negative 70 millivolts. Remember, we're just hanging out at negative 70 millivolts when we're at rest. Nothing's happening. As the ion channels start to open and those positive ions move into the cell, if we hit the threshold potential, and that's here of negative 55, so if enough positive ions come into the cell that we get to negative 55 millivolts, an action potential fires. And that's this piece here. This is an action potential. This is the message that's being sent down the axon. And the message that is sent, or the way a message is sent in our neurons, is that the neuron is typically negative, and all of a sudden we get this jump up to positive 30, and then the channels close and everything resets and we go back to the resting potential. But it's this sudden increase of positive ions into the cell that changes the potential of the cell from negative to positive to negative. And that switch is our body's way of knowing, hey, there's a message being sent. I'm going to go through this quickly one more time, just so you understand a little bit. I know this is complicated. You'll probably want to read it in your book as well. But we start where the inside of the cell is negative. The outside, the fluid that surrounds the cell, is more positive. And when the cell is at rest, we're at a resting potential of negative 70 millivolts. The inside of the cell is negative compared to the fluid outside of the cell, which is filled with these charged particles, these ions that are more positive. And what's the difference? How much more negative is the inside of the cell than the outside of the cell? It's negative 70 millivolts. When a message is sent from another neighboring neuron, some channels open in the membrane. All those positive ions are attracted to the negative charge inside the cell. So the positive ions go into the cell, and the cell becomes more and more positive. If enough positive ions go into the cell that we reach this threshold potential of negative 55 millivolts, we get an action potential. When we have an action potential, what actually happens is all the channels open and all those positive ions go into the cell at once, and you get this huge, massive change in the charge from negative to positive. And then those channels automatically close back up, and the cell resets. The cell basically sends those positive ions back out. There's a pump in the cell. It's called the sodium-potassium pump, more than you need to know. Sends the positive ions back out, keeps the negative ions inside, and we get back to our resting potential of negative 70, and then we're sort of waiting again for another message. And this change here, this is our action potential. What happens after this sudden jump in voltage? Well, like I said, that section reestablishes its resting potential. So we have an action potential, all those channels open up, the positive ions move in. You can see you get this up to positive 30, back down, it resets. And then this axon is long, and so we gets kind of like this message gets sent to the next area. And now these ions, you can see now we're going to move down here. Now these ions, channels open, and we get another action potential. And then that action potential, this resets. And then farther down the neuron, we get another action potential. And so you can see it's almost like if you were to light a fuse and you see the lighting of the fuse go all the way down. That's the same thing with the action potential. You get one action potential here, then it resets, and another action potential, and another action potential. And in that way, it gets sent from one part of the axon all the way to the end. So it's a cascading effect down the axon, with the ion channels opening and closing at each part, letting the message be sent. We call that spreading activation. Now, the message that's sent can be two main types. It can either be an excitatory signal. This is going to tell the neuron next to it to fire an action potential, like, hey, I saw something beautiful and I'm trying to get that visual image to my brain. And so each neuron is going to try to send this message. She saw something, she saw something. And so an excitatory signal, when it gets to the end of this axon, is going to say to the next neuron in line, hey, you need to make an action potential. please be sure to fire. There are also inhibitory signals, and those are signals that instruct the neuron after it not to fire, because we need to stop things as well, right? If I'm trying to bend my arm, I need to bend my arm and send the message to all the axons that are needed to bend my arm, but then at some point I need to stop bending my arm, right? And so I need inhibitory signals to now be sent saying, okay, her arm is bent enough, now stop sending those signals. And so the signals can be inhibitory or excitatory, and it's based on the neurotransmitter that's sent, and we're going to talk about that in a second. So for an action potential to occur, you've got to have more excitatory signals than inhibitory signals. There are typically many axons all communicating together, and so it's an additive process. If one neuron is getting 10 signals that say fire, excitatory, and 5 messages that are inhibitory, well, it's probably going to fire because the excitatory signals are outweigh the inhibitory. But if you're getting, there are 10 neurons around you and 10 of them are sending inhibitory signals and maybe 1 or 2 are sending excitatory signals, the cell will then not fire. It will be inhibited. So it's this additive process. In terms of action potentials, it's an all or none principle, meaning the neuron either fires or does not fire. There's no such thing as a small action potential or a large action potential. You either get one or you don't. And the key is, if in the beginning you hit the threshold potential of negative 55 millivolts, you're going to get an action potential. And if you don't, if only a few positive ions come in that you don't quite get to that negative 55 millivolts, then you just won't get an action potential. That's the idea of all or none. So if action potentials are all the same size, how do we differentiate between weak and strong stimuli? How do we differentiate between, someone tapping you on the arm and someone punching you in the arm, right? So neurons fire more frequently with strong stimuli. And then their neurons deliver their message to more neurons with strong stimuli. So if you're feeling a weak stimulus, then maybe you're just going to get one action potential, another one over here, another one over here. If you're getting a strong stimulus, you're going to get 1, 2, 3, a lot of them quickly, if that makes sense. Now, there is another aspect to communicating within a neuron called saltatory conduction, and that has to do with the myelination or a myelin sheath. The myelin sheath is a fatty substance that covers and insulates the axon and makes transmitting your action potential quicker. So the myelin, and I have that here it is, here's a picture of these myelin sheaths. They're actually made, this myelin sheath is made by glial cells. Remember those supporting cells? And so there are glial cells called oligodendrocytes, and they have these almost look like arms that come out and wrap around the axon. You can see those yellow axons, creating this myelin or this fatty patch that covers this neuron, this axon. In other parts of the nervous system, you have things called the Schwann cells. These are also glial cells. They actually wrap their whole body around the neurons, and this is in your peripheral nervous system, which we'll talk about soon, to create myelin, these patches of myelin. And so, here's a real life picture of these oligodendrocytes under the microscope, creating these myelin sheaths, these patches of myelin that cover and insulate the cells, axons. what happens there is when the cell is myelinated, the speed of impulses increases from less than 1 meter a second to about 10 meters a second, which is good because really what we really want is fast communication within our nervous system, right? If we want to send a message to bend my arm, I want that to happen quickly. I don't want to have to wait a long time, right? Or someone I see, I don't know, a fire in my backyard. Well, I want to see that and be able to run quickly. I don't want slow transmission of these messages. So myelin increases the speed. They use the word saltatory conduction, meaning that this action potential basically happens only at these uncovered sections of the axon. The myelin makes such a strong insulation that once you've got this action potential that's sent here, and then it just passively, the message is sent all the way down till you get to this next unmyelinated section. And when you get here, then you have the ion channels opening up, the positive ions moving in, and etc. There's another action potential. And then again, that message is passively sent quickly and passively down to the next unmyelinated section. And these little unmyelinated sections are called the nodes of Ranvier. That's where you get your actual action potential. And you can imagine that the process of making an action potential takes time, right? We were over here. You've got to have ion channels open up, ions move in, then the ion channels close, it resets. That whole process takes time. And if you had to do that up and down the entire axon, that would slow this whole process of transmission down. But as you can see here, if you can, now you can tell these are your myelinated sections, these little blue sections. You get an axon, and then it is you get an action potential, and then it is passively transmitted down the section that's myelinated, and then you get another action potential, passively transmitted. It really speeds up communication. So this provides rapid conduction of impulses and also conserves energy for the cell, because as you have to open ion channels and close ion channels, that takes energy. And of course, we want to minimize energy whenever we can, so we can save it for what we need it for. Multiple sclerosis is a disease in which the myelin sheath is destroyed, and these sections of myelin start to fall off. They basically die, and that's associated with slower impulses, poor muscle coordination, etc. So multiple sclerosis, which you may have heard of, is really just a disease of myelin. All right, so how do we get a message sent from one neuron to another? We said the neighboring neuron sends a message to the neuron next to it. And these neurons don't actually touch each other. There's a little tiny space between them called a synapse. So the synapse is the space between two neurons. It's the end of one neuron, right? The terminal, axon terminal of one neuron, and the dendrite of another. And we have to send a message between the two. And the message that's sent is chemical. Remember, we said this was an electrochemical process. So this is a chemical message that is sent. And this is what happens. Here we have the end of our first axon. So we had a bunch of action potentials coming down the axon saying, ooh, we have a message to send. And it needs to send it to the dendrite of the next neuron in line. And you can see we've got the space here called the synapse. Well, if the neurotransmitters, these are chemicals, are stored at the end, at the terminal of the axon in these little balls called vesicles. These are all filled with chemicals. And when the action potential reaches the end of the axon, the axon releases the neurotransmitters. You can see these vesicles actually start to move and they open up and the chemicals that are inside are released. And here's a bigger picture of that. Here's a vesicle that has now merged with the end of the axon. And these little neurotransmitters float across the synapse. It's a fluid-filled synapse. And they're looking for receptors on the other side. Here are receptors on the dendrites of the next neuron. And so as those chemicals float across, they bind to the receptors. And that is the message that's sent. The message that's sent saying, hey, I need you to send a message, are these neurotransmitters that float across and bind to receptors on the other side. And so that's the way that a message is sent. And you can see it here, that we've got an action potentials coming down the axon till we get to the axon terminal. There are neurotransmitters or chemicals in these vesicles. When the action potential reaches the end of the axon terminal, the vesicles start to move. The neurotransmitters exit the vesicles. They start to kind of float across the synapse, and they bind to these receptors on the dendrite of the next cell. So the neurotransmitters are the chemical messengers that use neurons to communicate at the synapse. And they're located in the vesicles, as I said. We know at least about 100 different types of neurotransmitters. The receptors are the location on the receiving neuron's dendrite where the neurotransmitters attach, kind of like a lock into a key. And then I'm going to talk about this process of reuptake in 1 minute. It's the process by which neurotransmitters are reabsorbed by the sending axon terminal. So again, here's another kind of schematic that we have the axon here, right? They're all lined up trying to send a message. The action potential is coming down the axon till we get to the axon terminal, which is the end of, we call this the presynaptic neuron. Well, now that should make sense because the synapse is this space. So the presynaptic terminal is the end of this neuron, and we have neurotransmitters in these vesicles. The postsynaptic neuron, that's the one that's receiving the message. This has these receptors on it. The membranes face each other through this fluid-filled area called the synapse. Once the action potential reaches the end of the axon, these vesicles start to fuse and the neurotransmitters spill out into the synapse. And then they start to bind to the receptors. And it's this binding that then says to this next neuron, hey, send an action potential. And now we get action potentials coming down this axon. And in this way, a message is sent from one to the next neuron. But you can see it's very complicated. It's not just, one axon and one neuron in line with another neuron. Oh, actually, here you can see the little vesicles, right, at the end of the axon. But there are many, you can see here, many axon terminals synapsing on one neuron. And this is where we said that it's really an additive process. How many of these are sending excitatory messages? How many of these are sending inhibitory messages? Last part, this is again the same idea. Now we've got axon A is our presynaptic neuron. It's sending a message. We've got action potentials coming down here, trying to send a message to axon B. We have our vesicles. When the action potential gets down to the axon terminal, the vesicles merge and the neurotransmitters spill out into the synapse, and they start to bind with receptors on the next axon or the next dendrites. Now, this process of reuptake means we also have to reset the synapse. We can't have neurotransmitters just hanging out in here because a message is sent, the neurotransmitters are released, and then we need to clean up this synapse. We can't just have random neurotransmitters in here. That becomes a problem. So the process of reuptake, it's almost like a recycling process where the presynaptic cell that released all of this neurotransmitter, some of it will bind, and that way we've sent the message to the next neuron, hey, you got to send an action potential, but some of it won't. Not every neurotransmitter will bind. There's extra. So this presynaptic neuron will actually take these neurotransmitters back up into the cell, reuptake. That's the process of reuptake. They take them back up into the cell. They can't use them again. They get basically recycled. But by doing that, we clean the synapse up so there's no neurotransmitter left, so that when another action potential comes down, we start the process again. In terms of neurotransmitters, there are so many of them. We're going to just go over a few. We have acetylcholine as a neurotransmitter. This is really important for relaying messages from neurons to muscles and for movement. So acetylcholine, muscles and movement. Too much leads to muscle spasms. Too little causes paralysis. So acetylcholine having to do with muscles and movement. It's also been implicated in Alzheimer's disease, that too little acetylcholine is seen in Alzheimer's disease. Glutamate as a neurotransmitter, you want to remember, is an excitatory neurotransmitter. It's one of the more common excitatory neurotransmitters in our nervous system and plays a role in learning and memory. GABA is an inhibitory neurotransmitter. So it slows everything down. It stops our body from sending messages. It plays a role in sleep and wakefulness, and particularly anxiety. Anti-anxiety medications cause an increase in GABA. And I just have a little side note here that many substances used to receive anxiety, like alcohol, cannabis, tranquilizers, use increasing GABA basically in your brain. So if you think about having anxiety or being very nervous, we've all felt this way at some point. What's happening in your body? Everything's kind of overactive. Your heart's beating fast. Your heart's beating fast. You're breathing fast. You're like maybe shaking a little bit. Everything's going so fast. And so GABA occurs naturally in the brain, and it has an inhibitory function. It slows down the action potentials. It slows down neuron firing. So without enough GABA, neurons fire too easily and too often. If you have too much GABA, you're too inhibited and you can't get moving. But many of the medications for anxiety affect the level of GABA and how it works in your brain. So benzodiazepines reduce anxiety through the way they interact with GABA receptors. Or, on the other hand, coffee inhibits GABA. It stops GABA from working. So again, GABA's inhibiting you, calming you down. Coffee stops that from working, and so it makes you more anxious. And so this is how a lot of drugs work, which we're going to talk about in a future chapter. Other neurotransmitters, norepinephrine, maybe you've heard of epinephrine, norepinephrine. This helps prepare you for stressful situations, get your body ready for fight or flight. So if you remember norepinephrine, fight or flight. And then serotonin, important with appetite, aggression, and mood. But low levels of serotonin are linked to depression. So that's another real big connection. When we're looking at depression, we see low levels of serotonin. And in fact, medications to treat depression often try to increase serotonin. All right, everybody. I know that. So let's see. We've got a couple more. Dopamine, important in attention. It's linked to schizophrenia as well as depression and substance use. Dopamine is really key in what we call the reward centers of our brain. So substance use increases dopamine quite a bit. And then endorphins, these are naturally produced opioids that are released in response to pain, increased in levels of exercise, but endorphins are like natural pain relievers. All right, I know that was quite a bit, but we're getting through this. The beginning of this class is always really heavy with research and very biology based. And as we move through the class, we're going to move into things like sleep and how do we learn and things like that. So good luck. If you have any questions about this, feel free to send me an email. I'm always here to help you. And I'll see you soon. Thanks.